Well, thanks to everyone for coming, and thanks to Matei for the shout-out this morning. So Tim and I are bio-curious mathware engineers from the Broad Institute of MIT and Harvard, which is a biomedical research institute located in the Cambridge that's next to Boston. And since this is the research track, I'm going to first describe some of the incredible challenges and opportunities in biology, and in genetics in particular, that led us to found the open source software project, HAIL, nearly 2 years ago. Then Tim is going to dig into the architecture, impact, and some of our future plans for the project. So we're actually growing as a new initiative at the Broad called the Initiative in Scalable Analytics, and that's located in the heart of biomedical research and technology, the Bay State. Now, the Bay Area also has a lot going for it in biotechnology and research, and especially in computer science. So here's the 8086 processor that led to the x86 architecture nearly 4 decades ago. And I think from this image of a square centimeter of silicon, we can all agree that computers are complex. They have hardware, they have software, many levels of abstraction for both, and there's execution happening at all levels. On the hardware side, we go from the physics of semiconductors all the way up to warehouses full of network machines. But if computers are complex, then I think it's fair to say that biology is ridiculously complex. Instead of silicon hardware, computation is performed in wetware, and that wetware is understood in terms of molecular biology, which is in turn understood through biochemistry, which of course is just simple quantum physics. And at the top of this stack, there are programs like taking sensory inputs to behavioral outputs or displaying disease phenotypes like diabetes or heart disease. At the same time, lower in the stack, you have 37 trillion cells in your body, and they're all running in parallel, executing programs like mitosis, differentiation, or immune response to literal viruses. And the ultimate source code at the bottom is your DNA, which can be corrupted by cancer. And the hardest part here is that while we designed computers, biology designed us through millions of years of evolution, and it didn't really give us an instruction manual with the architecture. So we're left with this very hard recursion, which is we have to use our brain to build tools to understand our brain. But we have made a lot of progress. So in the last century, we've understood the central dogma of molecular biology and the genetic code, which tells us how genes encoded in DNA are transcribed into RNA, which then is translated into chains of amino acids, which then fold into proteins. And these proteins could have one or many functions. This protein is called PCSK9. It's been in the news a ton the last few months, and I'll come back to why in a few minutes. Okay, so it's really complex, so why bother even trying to understand it? I think there are 3 main reasons. The first is science. We seem to have evolved an urge to add to the repository of human knowledge. It's fun, it's exciting. Technology. We can actually harness the computational power of biology to create new technologies. So, for example, nature invented reinforcement learning billions of years before AlphaGo came along at DeepMind. And we can now use advances in gene editing and directed evolution to concentrate millions of years of protein evolution into a week to create new antibiotics, entirely new materials. I mean, this is literally hijacking viruses and bacteria in order to do computation in biomolecules at a rate that far surpasses any supercomputer. It's like completely nuts. And then finally, we want to live forever. And in the Bay Area, I think Calico and the futurists are all over this mission. In the Bay State, we do frame it a bit less disruptively. We say we'd like to decipher this wetware and software stack of human biology in order to prevent, diagnose, and treat disease. All right, this is a really hard problem. Our understanding of most diseases is primitive at best. We have billion-dollar clinical trials. They usually fail. And in some classes of disease, like psychiatric disease, we haven't really had fundamental new classes of drugs in decades. And pharmaceuticals will not even begin to invest in these areas because they just don't know where to look. And so I want to convince you that's where genetics and engineering together can really help. So as a refresher, genetics, we have 23 chromosomes, 2 copies, one from our mom, one from our dad, and each set has about 3 billion digits in base 4. About 1.5% codes for 20,000 genes, And the other 98.5%, we're still just beginning to learn what it's for. And if we zoom up to the top of the stack, the 7.5 billion people, it's the case that actually any two of us are 99.9% the same. This makes us human. On the other hand, the differences between us are enough to really recapitulate human history just by looking at DNA alone. So here, these 200,000 dots each represent a person, and the clusters correspond to different ancestry groups, like African and European. This is, for example, three-dimensional, just PCA projection of your genome. All right, so this is pretty, but it's not just history that we're interested in. We're interested in disease, and the differences we have genetically actually impact our risk for disease. And that's because nearly all non-communicable diseases are heritable. Some of them, like cystic fibrosis, sickle cell anemia, Huntington's, these are caused by a single letter that's off. Others, like lung cancer on the left, are largely driven by the environment, smoking in this case. But for most common diseases, there's a complex genetic architecture, and that means there are many places in your genome that impact your risk. And here I've bolded, these are just some of the diseases that impact my own family, right? So these affect a lot of us. And two of my grandparents had Alzheimer's, 3 with heart disease. So looking at this, it's one motivation for why I moved from doing theoretical mathematics at MIT to doing applied math on biology through software at the Broad. Okay, so back to PCSK9, that protein. Here's a story from the New York Times, just a few months old, and it's talking about a $1 billion clinical trial of a PCSK9 inhibitor. And it found that it massively lowered cholesterol and significantly reduced the chance that a high-risk patient would have a heart attack or stroke. And here's the data. This is in the New England Journal of Medicine. What you see is that over 3 years of the trial, you see the difference in LDL cholesterol between placebo and those on the drugs, okay? And so looking at this, the question is, how did Amgen know, out of all the proteins in the genome, this was the one they should really invest in developing a drug to inhibit? And a big part of that answer is statistical genetics. So here we see the data for 2 genes across people with high cholesterol or low cholesterol. And when you look at the first gene, Sonic Hedgehog, that's actually a gene name, what you see is there's about the same rate of rare variation or mutation that's inherited in the high cholesterol and low cholesterol groups. But when you look at the second gene here, PCSK9, we see that LDL, low LDL, correlates with more mutations. And this suggests that perhaps if we could knock out that protein, we would lower cholesterol. And that's a hypothesis that can then be tested in a lab by editing a mouse, for example, and then taken into trial. And that's what happened. And in this case, we came up not just with a deep understanding of the functional role of this protein, but also now a new drug that could potentially help millions of people, even those without these rare variants. Okay, so by using genetics to inform where we should look, we would hope that this story can just repeat for every disease, every day. And that's because while the theory of statistical genetics goes back a century to Fisher, it's only today that we finally have the data. Okay, so most of the genetic data that we've collected in the world was collected in the last year. And here is a plot from a paper called, I think, Big Data, Astronomical or Genomical. It's arguing that very soon we'll stop using astronomical as the adjective for big data. It will be genomical. And the reason is because you can see on a log scale here how the data's grown since 2001, when the first map of the human genome was completed. That was about 15 years ago. It was $3 billion. And today, you can get your genome sequenced for about 1,000. In a few years, it'll be 100, maybe sooner. And if you project out this red trend line, that's doubling every 7 months, we get to everyone on the planet within a decade. Now, that dominates the blue line. The blue line is Moore's law, doubling every 18 months. And that gap is really important. We'll come back to it. And the fact is that costs are continuing to plummet because technology is advancing and also because demand is exploding, not just from hospitals and industry, but from biobanks run at nation scale, from academia, also from disease consortia that raise money to collect a lot of patients with a particular disease. And you may have heard, for example, of the Precision Medicine Initiative. That's a project to collect 1 million Americans' DNA, sequence it, and also all their quantitative health information. At the Broad, this is what our data looks like actually 6 months ago. Our internal rate of doubling is about once every 8 months, so these numbers are actually probably much larger already. And I should say that most of this data is sequencing human genomes, but these sequencers have become so high throughput that they really have become the 21st century microscope. And so we're measuring higher in the stack, things like gene expression and metabolites through DNA. And it doesn't end there. We measure the microbiome in your gut. We sequence tumors to understand which radiotherapies, chemotherapies, or immunotherapies might be best for the patient. And with gene expression, we can actually now measure not just in bulk tissue and average, but we can measure at single cell resolution how much each gene is being expressed. So researchers actually project that probably within a decade, the amount of data created through genomics will dominate not just other fields of science, but also tech. And I actually wonder if there's any way we can store that much data without literally putting it back into DNA. So we'll see what happens. All right, so I think now I've tried to convince you that like particle physics and astronomy before, biology is firmly now in the era of fourth paradigm data-intensive science. And that means instead of having a simple hypothesis and collecting a little bit of data to test that hypothesis, what we actually do is we measure everything, and then we run computational experiments on the data. And these experiments consist of three phases. There's deciding the scientific questions that are relevant and mathematical models that might make these amenable to computation. Then you have to implement those models, and then finally, you need to run them. And the results can tell us what's the next computational experiment to do, or maybe what's the next wet lab experiment to go do to collect data that will be very helpful. And it's a really beautiful cycle of learning, but there's one major problem. It's that gap that we saw between the red line and the blue line. Because we set out, right, to understand the biology of disease through high throughput measurement, but the scale and the complexity of the data is rapidly transforming what was a biological engineering problem into a software engineering problem. Industry is often too removed from the science to know the right thing to build. And in academic research, where funding and tenure are tied so closely to publication, much of the software consists of grad student scripts that are single core, untested, and maintained only until graduation. And this is the result. The result is a failure to scale. Implementation time has exploded due to a lack of good domain abstractions for expressing complex queries, the need to hand shard data, and then babysit hundreds of HPC jobs and restart them when they fail. And really, that's the best case scenario. That's when your problem is naively parallelizable. On runtime, we're well past talking about the difference between a second and a minute. Now it could be a minute, a day, or never. And so tragically, just as we've entered this golden age for measuring all the stuff of life, we've crippled our scientific imagination to ask the questions we'd like to ask of the data. Okay, so what do we need to do? Right? We want to generate a return, because this is a massive investment. So we need software that is powerful and flexible to express deep scientific questions as computations. We need domain abstractions that raise the conceptual level of implementation much closer to scientific discourse. And we need to give researchers a knob so they can turn up the compute to get the results back instantly with a negligible increase in cost. And we should do it in the open, as openly as possible, so that we can really iterate on new ideas, share them as a community, and support open source, reproducible scientific research. If we do that, we can drive down the latency of computational experiments and drive up the rotations per minute on the metaphorical engine of science. All right, so now that I've laid out this very modest vision of what we need to do, Tim is going to tell you how, with the HAIL project 2 years ago, we set out to do this, starting with the domain of statistical genetics.
So John has now told us how we're going to cure every disease and live forever. But let's come back down to Earth. What do our data actually look like, and how do we use them? Well, we have a matrix, and in this matrix, the columns are people. All of one person's genetic information is one column. And the rows are locations in the genome, or loci in genetics lingo. And one row contains all of the genetic information among all individuals at one locus. And the cells, even though I say matrix, are not numbers. They're structured, they have a complex schema. And sequencers are noisy, probabilistic instruments. So in addition to our best guess of what the DNA bases are, we also have metadata about the sequencing process that is essential for quality control and analysis. But our story doesn't end with just the genomes. If we actually want to use genetics to address disease, we need to line up the genomes with disease status. So we collect lots of information about individuals, like whether they have a disease, clinical measurements like LDL cholesterol, age, ancestry, sample cohort. And increasingly, this table is actually starting to resemble an entire electronic medical record. And likewise, we have lots of information indexed by position along the genome. We want to know if a given location is found in a gene, which gene, or if it has a predicted effect on the function of that protein. So this is what we want to do. We want to connect the genes to the disease. We want to use genetics to link biology to disease, to connect what's going on at the level of the cells to human health. Now, this is the glamorous part of genetics, but it's actually not where the analysts spend most of their time. Genetics data, like many other types of data, is full of noise. Sometimes human DNA from saliva is contaminated by bacterial DNA. Sometimes sequencers produce patterns that look like real human variation but are actually just technical artifacts. And sometimes, half of your samples that are labeled female have a Y chromosome. It's actually questions like the one I've put here that analysts ask most often. They're combining several sources of data, and they ask questions like whether your sequencing quality is uniform across these various cohorts. If you have diabetes samples that came from a hospital that collected blood, and you have healthy controls that came from a hospital that collected saliva, you have a lot of work to do. So one of our goals was to make it easy for analysts to ask this type of question. Our team formed 2 years ago as an equity-free startup embedded in a nonprofit biomedical institute. And we spent the first few months not in front of a keyboard, but in front of the analysts. And we interviewed them on their workflows, their wish lists, their pain points, their frustrations. And we learned that questions like these are not only critical for science, but they're also a bottleneck due to the scale and complexity of the data. And when we started building, Spark and Spark SQL seemed like an obvious place to start. And we like Spark. Spark is scalable. It has great high-level programming APIs. It has packages for statistics and machine learning. So if we frame our problem in Spark, we might use data frames. We have 3 tables. We can encode each table as a data frame. One for the ID index table, one for the locus index table, one for the matrix, which we can represent by coordinate, or 1 row per cell. It's actually pretty easy to ask this question now. And I've written some pseudo-SQL here to join these data frames together and compute these statistics. But we finally found something we didn't like. We found shuffles everywhere. Our data sets today are measured in terabytes, and our job is to build for the petabyte scale data we're going to have in 3 years. But this implementation wasn't even scaling to gigabytes. So Spark was a great place to start, but vanilla Spark was not giving us the performance we needed. We had to build on top of it and tie in other open source technologies. Spark is built for a set of general use cases, but we have our own specific needs. And there's another problem. Scala and SQL are very familiar to people in this room, but they're very intimidating to computational biologists. So we needed a high-level API in a language they speak, like Python, and statistical methods relevant to their domain, genetics. So to make these methods efficient, we moved from a coordinate representation to distributing along the genome axis. The individual metadata fit in memory on a single machine for now, and they can be joined in with a broadcast. And we bundled the genome metadata with the rows of genotypes because we use the two together so often. So now we have an alternative distributed representation of our 2-dimensional data. What kind of methods do we actually need? Well, we need the same functional methods that Spark offers. But instead of needing 1 filter, we need 3. We need the ability to filter out cells, filter out rows, filter out columns. We need map, which can change the schema of each cell. We need reduce, and we can reduce either by row or by column. And these functions are useful for computing statistics from the genotypes, like average sequencing depth, per sample or per locus. And implementing these reduce functions is actually a fun exercise. Reducing by row doesn't actually require communication because we store all the data per row together. And reducing by column requires information from every partition. So the implementation of these 2 methods is radically different. We implement reduce by row as an RDD map and reduce by column as an RDD reduce. And these implementations are efficient because they avoid the shuffle that you'd need of reduce by key on the coordinate representation. But we couldn't escape the full shuffle forever. We need join. Joining by individual ID is free because all of that is local, but joining by genomic position is a big problem. And it turns out we do this all the time. We want to include all sorts of external genome index information in our QC and our analysis. We have machine learning models that predict the effects of every possible single base mutation in the genome. This is 9 billion rows and hundreds of columns. We have vast clinical annotation databases. We want to include information from other genetic data sets that we have. Now, a typical Spark join will shuffle everything. You want to join key 1 on the right with key 1 on the left, and you have no idea where either key lives in either RDD. So you create a new hash function which sends key 1 to partition 7, and key 2 to partition 1, and key 3 to partition 9, and you shuffle everything against this partitioning. Well, you can do a little bit better by being completely neurotic about the partitioning of your largest RDD. You never do any operation that strips the partitioner, so you always know where key 1 and key 2 and key 3 live in this larger data set. Now, any join is going to shuffle the smaller data set against this known partitioning. But we have a problem. We're comparing data sets of comparable size. Often both range into terabytes. And we have a second problem, which is that partitioning doesn't actually survive the write to disk. If you write a data frame to disk, Parquet will randomly reorder partitions when you pull it back up. So to solve these problems, we built the ordered RDD. Now, this is a generalization of Spark's range partitioner to give us shuffle-free joins with other ordered RDDs. It preserves the partitioner to disk, which involves some rather horrifying machinery to read back the Parquet files, which use reflection to access private Spark classes, pulling out the original file name, and then using regex to get the original partition number. This is kind of horrifying. Well, how do we actually eliminate shuffles here? Well, we may still need to use the network, but we don't need to shuffle to disk. We know the range boundaries of each partition on the left and on the right, and this can tell us the list of partitions on the right that overlap with any given partition on the left. So to compute partition 1 of the joined data set, we need to load partition 1 from the left and partitions 1 and 2 from the right. To compute partition 2 of the join, we need to load partition 2 from the left, partitions 2 through 4 on the right. I think you get the picture. And for our use cases, this join strategy has been between 10 and 100 times faster for our data sets. We also got something else almost for free, which was very fast interval queries. Spark is a great tool for batch processing, but you really can't beat a relational database for single-key query latency. Ordered RDD lets us keep all of this batch processing power, but it also cuts the latency of targeted queries. So consider a query for a small interval, like one gene, PCSK9. Given that we know the partition boundaries, we know exactly which partitions this gene overlaps. So to query this gene, we need to only load these two partitions, 3 and 4, from disk. We don't touch the rest at all. It's an operation that takes time proportional to the data kept, not the total data size. So to make this concrete, I have a 20 gigabyte data set on my laptop, and I can query very small ranges, 4 to 5 partitions, in about 100 milliseconds. So where are we today? Well, we've transformed the way that analysts in our group do iterative QC, and supported their analyses by rebuilding most of the standard tools they used as natively distributed algorithms in Spark. And as a result, they're no longer spending days coding their analyses and days waiting for them to come back from the Broad HPC cluster. They're spending hours coding their analyses in Hail and Jupyter notebooks running on massive Spark clusters with thousands of cores, and they're spending minutes waiting for the results to come back. We cut one common analysis from 2 weeks to 10 minutes. Now, even though Hail is an academic effort, it's not an academic exercise. Hail has already been used for dozens of studies at the Broad Institute, some of which have already been published in major scientific journals. And we've also seen widespread engagement from other academia and industry around the world. We've had open source contributors, and we're especially excited by our interactions with Databricks and Cloudera, who are helping to make Hail more useful for hospitals and diagnostic labs and pharmaceutical companies. But we're just getting started. We see a clear path to improving performance by about 2 orders of magnitude just in the next year. Now, some of these stepping stones probably look very familiar because Spark is doing the same thing with Catalyst and Project Tungsten. But beyond infrastructure, we plan to continue building out statistical methods for genetics. We've implemented most of the standard ones inside Hail, so now we're shifting our focus to using advances in machine learning to model genetic data in fundamentally new ways. But remember the mission, to decipher the biology of human disease. And genetics is not the only data that can help us. Each of the trillions of cells in our bodies has the same genome, the same source code, but they compile it into neurons and blood cells and skin cells. We can collect RNA profiles for hundreds of thousands of single cells in one experiment and learn the cell types by clustering. In fact, the Broad is helping to lead a global effort to create a human cell atlas of cell types which can be used to guide our disease research. And at the same time, we're starting to collect deeper and deeper phenotypes, like gene expression in every tissue in the body, or time series activity tracker data in patients with Parkinson's. And we'd really like to model all of this together. To do that, we need new tools. We need to be able to query tensorial structured data along any dimension. We need fast distributed linear algebra to fit our models. We need the models themselves. We need interpretable probabilistic models with scalable Bayesian inference. We need to predict and learn deep representations of our data. And to do that, we need help. We are a small team working on big problems, and we are actively looking to hire software engineers, which we hear are a Bay Area crop. But whether or not you want to leave the sunny Bay Area for the snowy Bay State, I hope that we've been able to convince you that your engineering skills can make a tremendous difference in biology and medicine. To learn more about Hail, come chat with us or visit our website, or find us at the Cray booth for a demo today and tomorrow. We also run a seminar at the Broad on the intersection of biology and computation. So if you're interested to learn more, check out the videos at broadinstitute.org/MIA. Thank you very much for your attention. We're happy to take any questions.
All right, for questions, I'll run the microphone to you.
Thank you. I'm Taro Saito, Treasure Data. I was actually on personal genome analysis.
Sorry.
The volume's broken.
Sorry. So my question is, what is the major pain point of using Parquet format as an intermediate data?
So Spark is great for using Parquet with data frames, but we use an RDD API. And using the data frame to write to Parquet and pull it back as an RDD seems very inefficient. So if we write a data set to disk that's about a gigabyte, we would expect that would take about 2 seconds to read back. That's how long it takes to cat that file. And using data frames, you can achieve that kind of speed.
Yeah.
But using RDDs, it takes almost 5 minutes. This is somewhat disturbing, and we want to fix that.
I see. So what alternative are you trying to create? So yeah, actually we're trying to create a new data format other than Parquet for efficient output or writing to disks.
So we're certainly using Spark as a model, and the work that Spark has done with Java Unsafe has produced some pretty incredible performance. So using a binary row store in that case can get us pretty close to where we expect things to be. It's certainly at the prototype stage now, but maybe in a year or so, we'll be ready for more widespread use.
Okay, I'll contact you
Yeah,
later.
come chat with us.
Hi. Did you guys look into bucketing to speed up joins in Spark? Because that's pretty useful for us.
So I don't think that strategy lends itself naturally to the structure of the genome. We have sometimes billions and billions of keys. I don't know much about Spark bucketing. I'd actually be interested to chat afterwards about that.
Any other questions? I've got one question. So Spark's going to have a cost-based optimizer with Spark 2.2. How do you think you could fit your join optimizations into that cost-based optimizer?
This is kind of tough. So we have a matrix. This is the fundamental problem. That means we can't use data frames and data sets. So we have our own sort of cost optimizations and ASTs to try to compile ourselves. And as much as we can, we'd love to fit that in with Spark. But up until now, it hasn't been easy.
All right, one last question.
If I understand correctly, what you presented was all single base changes. Is this also applicable to insertions and deletions?
Yeah, so the VCF format, which is the most common format for sequence data, will capture both single nucleotide polymorphisms, these single base changes, but also short insertions and deletions. And so all of that is retained in our data representation. And actually, we're agnostic to the format. There's many other file formats for sequencing that you can bring in. For larger structural variations, there isn't really a consensus in the community about how to bring that into the standard formats that come off the sequencers. And so we are definitely thinking about that intensely with collaborators within the Broad and at other places. So at the moment, we don't have a representation for large sort of copy number variation, big inversions, and things like this. It's something on our roadmap.
All right, let's thank John and Tim once again.